I'm here today to talk about how to manage Mosaic results back to the patient. So there's been a huge amount of discussion in the last 2-3 years around mosaicism and do we want it, how should we report it, what does it mean from a patient management, and there's been lots and lots of people very, very scared, and how do I communicate this back to my patients, what does it mean? Well, thankfully, most IVF patients are reasonably well educated, I would say, but if we think about people in general, people live off images. They like stories, they like pictures, it helps them understand. So, if you was to speak to somebody and say, do you know what a mosaic is? Hopefully they would recognize a pattern such as this, where it may be a mosaic on the floor or the wall, but they would instantly know that a mosaic is something which is made up of different types to make a bigger picture. And this is basically what a mosaic embryo is. So hopefully, having got the patient to understand that this embryo is a mixture of certain components, we can move on to the next bit of imagery, and this is all available in our guides and available to help you. But when I started in this field 10 years ago, we were doing day 3 biopsy or polar body, and it was very black and white. You cannot have mosaicism with a single cell. So we had a very nice clear story, euploid good, aneuploid bad, easy patient message, let's move on. With the advent of NGS testing and the extra sensitivity around that, we're now starting to pick up these mosaic embryos, which weren't one thing or the other, they were in the middle. And this is what a mosaic embryo is. It's a mixture of good and bad. Now, from a patient perspective, this is a mosaic embryo on the right, and the pointer doesn't work on the screen. And we are, just to be sure that the patients understand it, biopsying the trophectoderm, which goes on to form the placenta. We're not touching the baby, so we're making the assumption that the trophectoderm is representative of the inner cell mass, the baby. So what does mosaicism actually mean? Well, this is very much a mathematical model. But the earlier a cell division goes wrong, the higher the potential for a number of abnormal cells to be left in the embryo. So if we have one of the cells error between going from 2 to 4 cells, if that replication carried on throughout the embryo to development, day 5, day 6 biopsy, that's going to be a 50% mosaicism. The later those cell divisions happen and go wrong, we end up with lower and lower levels of aneuploidy mosaicism. From a technical perspective, from a clinical perspective, you're really looking at 25% and above to be able to detect it. These late mitotic errors would still be scored as euploid. So really, we're looking at these early mitotic division errors resulting in mosaic embryos. So, from a patient perspective, they've got the result back, they've got a mosaic embryo. Is it me? Was it my eggs? Did I eat the wrong thing? Did the fertility treatment go wrong? Am I in any way to blame for having a mosaic embryo? And the answer to that, as far as we're aware, is no. So, there is no correlation that has been discovered between age and mosaicism, or indeed any kind of patient sort coming in. Now this is total mosaicism, this middle green kind of colour, and that includes embryos which have an aneuploidy as well as a mosaicism, rather than just a single mosaic event. But from clinically relevant mosaicism, age is not an impact on that. Another question a patient might ask: what if we do another biopsy? Do I get a better result or a worse result? Will it prove anything? Is it worth doing? So what this study managed to prove is, so they took an initial biopsy as the reference, and then looked at some extra trophectoderm biopsies, and then also a biopsy of the inner cell mass to see if the the first trophectoderm biopsy was indicative of the inner cell mass status. So, if the first biopsy was aneuploid, complete aneuploid, the inner cell mass was always aneuploid. So even though very occasionally they found euploid tissues, basically if your first biopsy is completely aneuploid, then there's a very, very, very high chance that whole inner cell mass will be aneuploid. That's a nice message for patients. Again, a complete segmental. So this is a subchromosomal change. This occasionally, again, high percentage, let's keep feeding back, will give you sometimes euploid results, but again, the inner cell mass is completely aneuploid. Then we get into the mosaics. So a mosaic complex is 3 or more chromosomes showing a mosaic result. Now, if you keep biopsying a mosaic, by definition, a mosaic is a mixed population. So you have an increasing chance of finding a euploid, but again, the inner cell mass is very rarely euploid. Once we get to the single chromosomes, then the biopsy, you have a much higher chance of finding some trophectoderm, and it's 50-50 really whether the inner cell mass is normal. That's all very well, but so what that is basically showing is if you do another biopsy, you're going to get the same result. It will still be a mosaic. The level may change subtly, but you're not going to clarify that it was just that cluster of cells that gave the wrong answer. So in terms of managing patient expectations, talking to the patients, messaging them, this again, we've got all this in our publications over on the stand over there. So basically, if you have a euploid embryo, your patient, your clinic cohort is going to have an effect on that, but the published rate of a successful pregnancy after a euploid transfer ranges anywhere between 50 and 70%, depending on the clinic patient cohort. If we go to mosaics, which is how we started off as a company, as just reporting anything within a range as being mosaic, you get a 41%, so that doesn't seem so bad. But then when we started looking in more detail, then basically it's what we call a low-level mosaic, so between 20 to 40%, their implantation rate isn't actually that different to a euploid. And this is about the level that we could see with the technology on array CGH before we moved to NGS. Anything below 40% was called euploid. But there is a clear statistical jump between 40% and above. So if you start transferring highly mosaic embryos, which are closer to abnormal than normal, then your ongoing pregnancy rate drops to 22%. And if they're really complex mosaics, so lots of chromosomes going wrong, then they have a 12%. That's halved again. So what this means from a patient perspective is they have a chance, it's just a reduced chance. So we moved on to these reporting to help the clinics, where if you're in the US, you get the sex, the gender of the embryo. If you're in Europe and many other countries, that's not allowed. But just to show that we could do it if you wanted to. And then you get a kind of euploid normal, abnormal, low-level mosaic or a high-level mosaic result. But that, at the end of it, is there for a prioritization tool. So one thing that's been a lot of discussion is what happens to aneuploid or mosaic embryos. Should you discard them? Should you transfer them? What should you do? Well, you should prioritize your embryos. So highest priority is a euploid, clearly. Your lowest priority is an aneuploid. There are published papers of people transferring what have been scored as aneuploid, but generally they're not transferred. But then you go to a low-level mosaic and then a high-level mosaic as a preference. But the other thing I think which is really important to mention is that this isn't the end of the journey. So with the increase in vitrification success, the increase in the the applicability, and we've got lots of vitrification stuff on the stand if people want to come have a look at that. With good vitrification cycle and process, you can bank that embryo and have a repeat cycle and come back to it if there isn't any euploids generated. To get really into it for the embryologist or the person facing the patient, we also have this guide that just, and again there's some copies on the stand over there. Even within a Mosaic embryo, there are guidelines about which ones to transfer. So most people would avoid a Mosaic 13, 18, 21 and the sex chromosomes because they are the highest risk of a live birth, an affected live birth, if that mosaicism continues, whereas your highest priority is chromosomes 1, 3, 10, 12 and 19. You would always do these transfers with genetic counselling, of course, and making sure. But really, this is a short talk. So what we as CooperGenomics see this as a partnership with the clinics, with you guys, with the researchers, and we're always looking for information back from the clinics that we work with so that we can report based on the latest evidence that is available. And it will change. Mosaicism didn't exist 2 years ago. I'm sure the guidelines will change. But we believe in constant education of the clinicians and the patients, and we will do clinic visits to help with that. It's not just a phone call. So again, we have this clinician's guide to mosaicism on the stand over there. So with that, I will wrap up my talk, and I'm happy to answer any questions.